Good morning, students. Today I am going to discuss session 9 of chapter 6 from class 12 biology syllabus. The name of the chapter is Molecular Basis of Inheritance. Already we have completed 8 sessions from this chapter, and from these 8 videos we came to know a lots of things about molecular biology. So first thing is the different experiments on genetics, different experiments on the genetic material. The second thing is the structure of the nucleic acid, function of the nucleic acid. Third is the Watson-Crick DNA model. Fourth, DNA replication. Fifth, transcription. Sixth, genetic code. Seventh, translation procedure, and the last one was lac operand concept. So in today's video we are going to discuss the genomics portion. Today actually we are going to discuss the human genome project. Okay, so this portion actually belongs to the genomics portion. So first actually you have to know what is the basic meaning of the genomics, what is the basic definition of the genomics. So this is the study of genomes. The genomics means the study of the genome. Okay, so it is the study of genomes through analysis, sequencing and mapping of genes and non-cystonic areas with the study of their function. So this genomics term actually was coined by scientist Thomas Roderick in the year of 1986. So US government started this human genome project in the year of 1990, which is coordinated by the Department of Energy and the National Institutes of Health. So first you should know what is the basic definition of genome. What is the definition of genome, or what is the meaning of genome? So this genome actually the whole hereditary information of an organism that is encoded in the DNA is called the genome. Okay, so in our body lots of genes are there. Okay, so many genes are there. So this is collectively called the genome. The whole sets of gene in our body, the whole organism's body is collectively called the genome. Okay, so the whole hereditary information of an organism which is encoded in the DNA, that's called the genome. So this human body actually made up of about 100 trillion cells grouped into over 260 types. So every cell contains the same number of chromosomes and the same kind of genome also. Okay, so what is the meaning of human genome project? So this human genome project actually was called a mega project. This human genome project was called a mega project because this whole project actually started by US government. And what is the aim of this HGP? What is the main of this human genome project? That actually it has total triple aim. Okay, three aims are there. The first aim is the mapping the whole human genome at the level of nucleotide sequence. Okay, so human genome have approximately 3 into 10 to the power 9 base pair. So the cost of sequencing is required $3 per BP. So then total estimated cost of the project is $9 billion. So that's why this belongs to the mega project. So why this is called the mega project? Because it needs more money. It's need more money. So then total estimated cost of the project is $9 billion. Okay. Next thing, why this is called also the mega project? That they obtain. So actually the obtained sequences were to be stored in typed form in books. Okay, so this is actually one data collection center. So if each page of the book contained 1000 letters, so each book contain 1000 pages, so then 3300 such books need to store information from a single human cell. So we need 3300 books needed to search this actually store these kinds of information. Okay, from a single human cell, and we have approximately 100 trillion cell. So can you imagine that how many books we require to store all the informations of our whole genome set. So that's why this belongs to the mega project. So this human genome project actually belongs to the mega project. So what is the aim or goal of the project? So the first aim, so this is important for your exam. So what is the aim of the project? First thing is to identify the approximate 22 lakhs to 2,05,000 genes in the human DNA. Okay, so to determine the sequences of the 3 billion bases that make up human DNA. Next thing, to store this information in databases. Fourth, to improve tools for data analysis. And the last one, to address the ethical, legal and social issues that arise from the genome research. So what is the actually the main aim? That the first aim is the mapping the whole human genome at the level of the nucleotide sequences. The second aim is to store all the information gathered from the project in databases and develop tools for the data analysis. And next thing, to solve any ethical, legal or social problems which actually may arise from the project. An international human genome project was started in the year of 1990. Okay, and the project was completed with the help of the automated DNA sequencing machines on June 26, 2000. Okay, so it needs approximately 10 years to complete all the projects. So next thing is the revelation of XGP. So what is the methodology? So first thing is that the methods involves two major approaches. So it involves the two major approaches. The first thing is the identifying all the genes that expressed as RNA. First thing is the identifying all the genes that expressed as RNA. That's called the expressed sequence tags. It's called the expressed sequence tags. Okay. Next thing is the blind approach of simply sequencing the whole set of genome. So that contained all the coding and non-coding sequence. So it contained all the coding and non-coding sequence. So later assigning different regions in the sequence with the function. So that's also called the sequence annotation. Okay, this is also called the sequence annotation. So this is the methodologies of the XGP. Okay, so this human genome contains about 3.2 billion base pairs. Fruit fly has 132 million base pairs. So this is 1/24 of the total length of the human genome. So this is just the 1% out of 24 of the total length of the human genome. Okay. Next thing is this XGP, this human genome project actually has found out that the human genome carries about 3 lakhs to 4 lakh genes. So the number is the same as present in the mouse and double the number of the genes in fruit fly. Next thing is the size of genome of number of genes is unrelated with the complexity of body organization. I am repeating again, the size of the genome of the number of the genes are actually become unrelated. Okay, so with the complexity of the body organization. So lily plant has 18 times more DNA than the human genome. In spite of this fact, the lily produces fewer protein than the human beings. Okay, so many types of the unrelated things arise. So eukaryotes also contain mostly the split genes, which genes are actually break. That's called the split genes, having the long non-coding areas or introns. So already we have discussed this intron portion in our transcription portion. So further, not more than 10% of the genome contains coding areas or exons. So the remaining DNA is non-coding, having either pseudogenes or repeated DNA sequences. Okay. So this human gene actually contains 3164.7 million nucleotide base approximately. Obviously it's approximate estimation. Okay. So the average gene consists of 3000 base pairs, but size actually varies. The largest known human gene, being dystrophin, has 2.4 million bases. The total number of genes is actually estimated at 30,000. It is lower than previous estimates of 80,000 to 1,40,000 genes. So almost all nucleotide bases are exactly the same in all people. Almost all nucleotide bases, this 99.9% nucleotide bases, are exactly the same in all people. Okay. The functions are unknown for over 50% of discovered genes. So less than 2% of the genome codes for the proteins. So here the repeated sequences make a very large portion of the human gene. These repeated sequences actually make up very large portion of the human gene. So the part of DNA actually which contains the repeated sequence, that's called the satellite DNA. I am repeating again. Please mark my word because this is important for your 1 mark question. The part of DNA which actually contains the repeated sequence, that's called the satellite DNA. So satellite DNA is of several types. So one of them is moderately repeated sequence of 150 to 300 base pairs, and they include transposons and ALU elements. So many satellite sequences are 11 to 60 base pairs long, hypervariable repeat sequences. So they are VNTers used in DNA fingerprinting. Over a million copies of simple sequence repeated or microsatellites occur in the genome. So they have 5 to 8 base pairs. So these repeated sequences are called the junk DNA. Okay, so I am repeating again. So what is the meaning of the junk DNA? That over a million copies of simple sequence repeated or microsatellites occur in the genome. So that's also called the SSR. So they have 5 to 8 base pairs, and these repeated sequences are called the junk DNA. Okay, so all those informations are actually important information for your 1 mark question. So chromosome 1 has most gene, 2968, and the Y has the fewest, 231. So scientists actually have identified about 1.4 million locations where single base DNA sequences, that SNP, already I have discussed that the single nucleotide polymorphism, that's called the SNIPS. So this information actually helps to finding chromosomal locations for disease associated sequences and tracing human history. I am repeating again. So these kinds of information actually helps this SNPs, this single nucleotide polymorphism, this all those informations actually helps to find the chromosomal locations for disease associated sequences and tracing the human history. Okay, so this is the main thing of the human genome project. So actually, so now you can ask one thing, that what is the prospects and implementation of this HGP? What is the implications of the HGP? So the first thing actually, the study the genes that will cause the reversion of the cancerous cells to normal cells. So studying the genes. So by these SNPs, by this single nucleotide polymorphism, by all those things, you will actually get to know that which sequence is diseased, which sequence is normal. So studying the genes that will actually cause the reversion of the cancerous cells to the normal cells. And it will be possible to study interactions between the various genes. So through the HGP actually it possible to study the interactions between the various genes. Next thing, it holds promise of healthier and longer living designer drugs and genetically modified diets according to the needs of the individual human beings. Next thing is the single gene defects produce a number of hereditary diseases. So their exact location, reason for defect and method of correcting the same can be found out. Okay, so by all those things, so through all those things you will get a better healthy life. You can get a better healthy life. So this is the main prospects and implication of the human genome project. So I am repeating again this all those human genome project. Why? Because this human genome project is quite bit difficult and quite bit tricky and unknown to all of you. Okay, so from this portion, first of all you should know that what you have to study. The first thing is the what is the basic things of the genome. So this is also important for your 1 mark question, that what is the basic things of the genome, what is the basic definition of the genome, how many genomes are there in our body, how many genes are there in our body. Second thing is the what is HGP, what is the meaning of this HGP. Okay, and why this is called the mega project. So you should know why this is called the mega project. So the first point and second point is there. I have written 2 points. The third things are the main aims or goals of the project. The next thing is the what is the aims or what is the goals of the project. Okay, so I have written total 5 points. So from these 5 points, within 5 points, just you will remember 2 to 3 points. Okay, next thing is the methodologies. What is the methodologies, what is the methods. Okay, so this is not actually important, so all those things are important. This what is the meaning of the ESTS, what is the meaning of the sequence annihilation, this portion. Okay, I am highlighting this portion, the sequence annihilation portion. So all those things are actually important. Okay, next thing is the what is the goal or what is the main revelation of the HGP. Okay, and the last one is the what is the prospects and implication of the HGP. And from this portion some terms are there, like I am writing, like the junk DNA, Okay, so all those things are important for your 1 mark question. Or this SNPs portion. Okay, this portion is actually important for your exam, that what is the meaning of the SNP. So all those things are actually important for all of you. And this is your last topic for today. In our next class we will discuss the last portion of this chapter, the DNA fingerprinting process. Okay, so these all those informations are quite bit new to all of you and quite important for all of you. Okay, and these things are actually quite boring, rather than the other topics, the transcription, the translation, that operon. But you have to know all those things, you have to know for all those things for your exam. So just follow me, that what I am saying, just follow all those things, just 2 to 3 points just you have to remember from this portion, and the aims and goals and the what is the prospects or implication of the HGP. And I am saying one thing, I am saying repeatedly, that these chapters is very much difficult chapter for all of you. Okay, because some actually maximum topics, maximum things are quite new to all of you. So that's why please go through each and every topics properly from your NCERT books first, and then follow all the videos which I have given, because all the videos will help you a lot for better understanding. Okay, and in the meantime, so if you are having any queries, any problems, just ask me immediately. Okay, so you have to know each and everything, and that the possible question what may be come, which type of question may come in your exam, not only for your school exam, for your others exam also. And this chapter actually quite difficult chapter. First of all, this chapter is quite difficult chapter and quite important chapter, not only for your school exam, for your others exam also. And I hope already you have gone through all the videos which I have given on the chapter 6. Okay, only one things are actually left, the DNA fingerprinting. After that this chapter will be completed, and after that I will start a new chapter. I will start to actually discuss a new chapter, that chapter 7. Okay, so please go through each and every videos properly. Okay, so in this video actually I have highlighted some portion, I have pointed some portion, some informations are also I have given. So I think all those things are helping you a lot for understanding, for easy memorization. Okay, but after that if you are having any problems, any queries, just ask me immediately. Okay, but go through these portions properly, go through this chapter properly, because minimum 17 marks question, 10 marks, minimum 10 marks question will come from this chapter, with short question, with very short question, with long question, with MCQ questions. Okay, so please go through each and every videos properly. Okay, so now I am ending this session. So in our next class we will discuss the last portion of this chapter. Okay, thank you, bye.